So welcome, everyone. Thanks for attending today. For anyone who's attending this call for the first time, we've been holding this industry-wide call every month with companies that want to contribute to the cultivated meat value chain. And so the purpose of these calls is really to offer new and existing cultivated meat stakeholders an opportunity to learn from each other with respect to some of the obstacles that they're facing, as well as some of the potential solutions that are being developed by others along the value chain. And so hopefully the end goal is to really help to accelerate research and development as well as commercialization of cultivated meat. So if your group is interested in presenting on a future call, maybe you're working with a company that could benefit from being on these calls, do feel free to put them in touch with me directly and we'd be happy to get them added. A reminder that you can also extend the invitation to other colleagues that are at your company in case they're interested in attending or getting updates from us via email on some of the important announcements that we make to companies in the sector. So just briefly, a couple of housekeeping items from our side. I've been in a lot of people's email inboxes with a request to participate in this survey questionnaire that we have currently live that will help us inform a new techno-economic modeling project that we've started at GFI. And the intention here is to really bias these models with real company data rather than what we can find in the literature for other cells or coming up with assumptions that may not be as data driven. And so you can help us with that by completing the survey. If you have information on cell composition or nutrient kinetics and that sort of thing. So I'll drop that in the chat. We'll be keeping this survey open and live throughout January. So if you can consider participating, it would be greatly appreciated. And so that's it from my side. Today, pleased to welcome Mario Nataro from CUBIQ Foods, who's going to be discussing the importance of fats as ingredients in the alternative protein sector. And then at the halfway point, we'll turn it over to John Barlow from Biocell Energetics. He's going to discuss how metabolic profiling can help to improve production efficiencies. As always, you can drop questions in the chat throughout. And Mario, I will turn it over to you.
Thank you, Zach. Thank you for inviting us here. It's a pleasure to talk at the Good Food Institute Cultivated Meat Seminar. I think it's a great way to share some concept and technology developed by young companies in the alternative protein space. And what I really like of this seminar, of this platform, which is actually a viable opportunity for collaboration and a meaningful interaction. And yeah, I'm excited to share what has been going on lately in CUBIQ Foods. So my name is Mario Notari, and I am a senior scientist and R&D coordinator at the Cell Culture Lab at CUBIQ Foods. Here are my contact details. Here is my mail, so feel free to drop me any question or go to contact me. And here you have the website of CUBIQ Foods. So today my talk will be about fats and how CUBIQ Foods technology are supported food producer, traditional and the new generation one, to adapt their products to the new era of food production. So here there is the outline of my talk today. So I will start sharing how the food and the way how the food is produced is evolving. And I will be focused on the challenge of that food producer are facing as they are adapting their product to the new ingredients that they are coming up. Then, of course, I will talk about why fats matter and what is their role in this in this revolution. Then I will quickly talk about CUBIQ Food vision and mission, and what are the technology that we are developing to help the food producer to adapt their product. And especially I will actually talk how our technology can actually help also the cultivated meat space. Then I will close with final remarks and take-home message. So the food industry is undergoing a significant revolution that lately, in the past 10 years, dramatically speed up. So the food and how the food is being produced is always been through an evolution. At the industry level, maybe not the customer perception, we can actually say that we already transitioned from a traditional production method to produce a food to an ingredient focus approach. I mean that historically food production relying on raising animals on the farm and then most probably processing them directly there and then consuming. So now the major food producer, they are producing the food on application are centered around the ingredients. In fact, the major food producers are usually mixing and blending different ingredients that actually come up with a final food. These change has been driven by the recognition that traditional methods are no longer sustainable in the long term, posing challenges to the global food accessibility, and then also about security. So in the context of ingredients, also the ingredient has been evolving during this time, and if in the context of the fats, also fats has been evolved. So as an ingredient, we actually pass from animal fat to vegetable fat. And then also this changing has been actually in parallelism with the customer's experience. At one point, also mixing different oils was actually seen as a sign of poor quality on the food. However, lately, the food that actually tastes very good is actually full of some of what we call refined vegetables that actually, yes, are vegetable oils, but are refined, and especially their sources are not very sustainable. And then, as we have seen in the last few years or so, we have seen that the major food producers are switching to new methods of producing the food, because the future of food will require technology to produce ingredients and also to use into the food. That's the traditional method and the traditional world. It was not being it didn't have. So in this vision, CUBIQ Foods for the future of foods is actually vision the emulsion that it will be a technology that I think it will be very important to food producer to use and adapt their food to new customers' preference. So in all this revolution and the next generation of food, we think that fats are always they have always been very important. They are important, of course, for a nutritional point of view. They are a dense source of energy. They are supporting a lot of cellular function. They are facilitating the absorption of essential vitamins, and they also provide critical fats that our body cannot produce, are omega-6 and omega-3, that are very important for the function of a lot of our organs in our body. However, well, fats are also playing an important role in nutrition. So we are not seeing fats as only an ingredient, but also as a macronutrient, and also gave us an opportunity to enhance the health profile of food product. Actually, the fats, they can be used to reduce saturated fat intake, so improving the health. However, in tasty food nowadays, we are actually seeing only what we call bad fat. However, we are thinking that fats are also a key player and a key ingredient for the functionality of the ingredient. In fact, fats are indispensable. They are contributing to a texture, to a flavor, to a mouthfeel, which actually been very important for customer acceptance. So we think that in the next generation of food product, particularly when we are talking about protein-based or alternative protein, replicating the functional property of these fats will be a significant challenge. And we need technology that help food producer to overcome these challenges. So we actually realized that there is a technology gap between the traditional food producer, fat as an ingredient, and the customer satisfaction for the product that belong to the alternative protein sector. The rise of alternative protein has created a unique intersection of opportunity and challenge for traditional food producer. In fact, while we have seen a booming in the protein sector, with alternative protein and cultivated meat, traditional producers often lack of the technological tools and expertise to actually introduce correctly these ingredients into the new generation of food. And particularly, this gap is particularly evident when it comes to fat. Producer, they always been used to use animal fat, but when they are seeing the other forms of fat, especially in the next generation, they actually don't know how to actually react. So that's why CUBIQ Foods is actually stepping in, or better, company like us are stepping in and are helping these producers to develop products that are more sustainable, are more tastier, and they actually are more nutrition. So CUBIQ Food is developing these technology that will help food producer. At CUBIQ Foods, we wanted to drive the revolution, a revolution that's in quality and sustainability of our food. We are actually commitment to transforming the global food industry by improving the health, the nutritional values, the sensory experience, and of course the sustainability of how we are going to produce the food. We think that we are an era that's high quality food product and never been more urgent, because we have a growing population and affinitive natural resources. So the food industry, they are actually facing immense challenge. Our vision is to deliver fast, forward-thinking solution, innovative solution based on scientific thinking that they can meet the needs of both producer and consumer. How we are going to do that? Well, we are pioneering. We are developing two technology, and the first one is called Go Omega 3, and is a pioneering technology for the introduction of omega-3 fatty acid into an everyday food product. As we know, omega-3 is an essential nutrient, is a well-documented health benefit, and with our technology, that is a microencapsulation technology, we are able to actually introduce it to most type of foods. But and we believe also that alternative fat are not, they can actually be used as a replacement to lower and replace animal fat in the plant-based world. So as a second technology that I would like to talk about a little bit more in detail, that we developed a couple of years ago, is called GoDrop, and we developed this blended formula that actually has vegetable oils and protein, with other natural components, which are usually polysaccharides, and which we structure as an irreversible gel that has the particularity to that we can integrate also different kinds of fat. So is a stable oil in water emulsion. They have a formulation that can be flexible, but and I'm going to talk this a little bit later on in this talk. What is really good that is actually this technology, the GoDrop, is actually a platform, and it can be tailor-made for many of the application of the different application that the food producer want. So in this way, we can help food producer to lower the saturated fat, to lower the energetic value, which has a pretty huge impact in the nutritional profile. As I said before, can actually be applied in several applications. So GoDrop is an innovative emulsion which contains hydrocolloid and is a fat replacer. It's been designed as a sustainable alternative to fat animals for food producer, but also for tropical and plastic fat. I'm talking about cocoa oil, palm oil, shea butter, and all these kind of oil. The formulation of GoDrop is made with regulatory approved ingredient and actually do not rely on any chemical process. Additionally, the GoDrop can actually ensure excellent physical stability during storage or during stress application, like could be a thermal heating as cooking. Is also offering a versatile and sustainable solution, and actually can be placed into the modern food application. So the GoDrop, like CUBIQ Foods, we actually realized that this technology could solve or could help overcoming a new challenge that actually cultivated meat sector is actually facing. And I'm talking about the first generation of cultivated meat product. Here I just depict, as you guys know, all a complete bioprocess for a cultivated meat product, and each of these sections, they have specific and technical challenges. So the company that they could complete a bioprocess, now they have a first generation of cultivated meat product that usually are well, actually depicted here, and they usually are still high in cost, however, that they lower it down the cost of media, etc., and most of the time we had a lack of structure. And these two characteristics make it very difficult to do 100% pure product, because it would be too much expensive. And having a lack of structure, let's say that for food producer is very difficult to introduce them into their food. So since GoDrop has a water and is an oil in water solution, and this oil part can be replaced with other sources of oil, actually can be cultivated fat. So in this way, this technology can actually be ready-made to, well, lower the quantity of the cultivated meat product, and in this term of cells inside the matrix, and most importantly, this matrix already has application in the meat, in the meat food application, but also into the plant-based world. Another new challenge, apart from the classic one that are affecting the cultivated meat industry, are a capex limitation and a problem of a specific expertise. Let's say that after 10 years of this cultivated meat is actually live, let's say that there are other problems. So the company that are just getting a finance, most probably they don't have the sources to actually structure a lab, and they will have to spend a lot of time facing problems that other company they might have already solved. This is also due for a lack of collaboration and partnering between the industry. So CUBIQ Foods' approach to this challenge is to actually offering his expertise and his technology to the other cultivated meat company. In these years, actually, we were one of the first industry to get substantial finances from the cultivated meat sector. We actually developed a lab-scale bioprocess to produce avian cultivated fat, and so we know very well what are the challenges that this bioprocess is. So during this journey, we produce the different technology which around the cell line development, and we were able to formulate 2 serum-free formulation. So here are just a few data on that. So we actually secure a easy method to isolate the different cells that actually could comply with the bioprocess requirement for cultivated to produce avian cultivated fat. So here there was the proliferation with a good PDT, and then they were able to accumulate fat. At the same time, we actually developed 2 chemically defined media, one for the proliferation of these cells and the other one for the differentiation. And as you can see here, by applying proteomics and design of experiment system, we could reduce the price, and we actually develop a completely chemical-free media for fattening of the cells. We are equipped with a state-of-the-art laboratory We have bioreactor, rocking motion, and the steering base. We have an R&D space, and we have a pilot plant space with a 200-liter bioreactor. And with this slide, I will finish the talk, and then with the final remarks that traditionally food producer have decades of experience in working with animal fats, but replicating their functional and sensory qualities in the new generation of foods that it become a challenge. And without this innovation, the products may fail to meet customer expectation. CUBIQ Foods is developing innovative technological solutions that help food producers enhance the quality of their product using the next generation of ingredients. This technology can also be applied to the cultivated meat space. And CUBIQ Foods is looking for partnership to share its cultivated meat technology experience, and as well as lab space and instrument to facilitating the advancement of the cultivated meat sector. And with this one, I will just finish, and happy to take any question if there is any.
Excellent. Well, thank you for that presentation again. I know it's difficult to do so in non-native language, so appreciate getting through that. And I'll just drop in the chat here too. I know you mentioned the omega-3 encapsulation, which is really important for the stability of those ingredients. And we actually have a report led by my colleague Claire, who's on this call, that talks a lot about this and how they might be pretty important for alternative seafood products in the future if people are interested in checking that out. So thanks again. And Johnny, we'll turn it over to you now.
Yeah, thanks, Elliot. I'll just share my screen. Yeah, thank you, Elliot. Thank you, Annie, for inviting me to give a talk today at the GFI seminar. And I'm Jonathan Barlow. I'm from the UK and currently recently founded Biocell Energetics. We specialize in using sort of real-time metabolic profiling to probe cellular energy metabolism in cells, looking at things like energy demands and capacities at a cell level in in vitro setup. And I'm really intrigued to know whether some of the technology we're using, some of the services and expertise that we have could integrate into some of the earlier parts of the supply chain for cultivated meat production. So I'll be really intrigued today to get any feedback from other people in the call today, and see if anyone else is thinking or looking into this. What we do is essentially we're a company spin out from the University of Birmingham. And we provide services in cellular bioenergetics for a fee, and we develop assays. We can design assays, analyze data, interpret data for our clients. And typically, we're working with academia. We work with CROs, other biotechs, startups to kind of support metabolic research, essentially. We design projects and manage them for research and development purposes, and we can also provide consultancy services and expert advice around cellular metabolism research. And our mission really is to increase the awareness of cellular bioenergetics, what it is, and how modern day technologies can be used to profile cells in real time, and how we map energy supply demands and capacities, and how this sort of mapping can help researchers and industries optimize and innovate or provide new innovations at the cell level. So we typically work in 4 different market segments: drug discovery, particularly looking around mitochondrial toxicity of off-site toxicities of compounds, and exploring new targets linked to cellular metabolism for drug development. We work with researchers in environmental toxicity, currently looking at impacts of things like microplastics, nanoplastics in water pollutants on human and animal health. We also work with professional sports, looking at developing sort of bioenergetic health biomarkers to optimize performance and recovery. And we support generic sort of energetic phenotyping for profiling cellular energy use in cells and tissues. So I think it's important to address what do we mean by cellular bioenergetics? What is it? So essentially, it's a study of how cells produce energy, how they manage that energy, and ultimately how they consume that energy for their functions and processes. Now, energy production typically is going to come from two main energy producing pathways, one being glycolytic flux and one being oxidative flux within the mitochondria of the cell. Cells will manage the energy and supply the energy to where the cell requirements are for it, for things like growth, protein synthesis, DNA synthesis. So the main pathways that we're interested in monitoring, or what we look at in the lab and with the technology we use, are looking at glycolytic fluxes and oxidative fluxes. So for glycolytic flux, this is what happens in the cytosol of the cell. We measure this through monitoring protons that are released into the media that the cells are growing or surrounded by through acidification changes. And we can map and calculate how many protons are actually being exported into the extracellular media. And glucose is the substrate that fuels glycolytic flux. It's glucose catabolism to lactate, essentially. So one of the byproducts of this pathway is obviously lactate generation, and you're also going to get changes in pH, which needs to be managed. Certainly, if we're talking about scaling cells that are very glycolytic, in terms of meat production or any types of cells that you're growing at scale in tanks, something that would need to be considered. The other thing we're measuring is the oxidative flux of the cell through oxygen consumption within the mitochondria of the cell, where oxygen is reduced to water at complex 4 in the mitochondrial respiratory chain. And substrates that fuel mitochondrial oxidative phosphorylation could be glucose, could be pyruvate, other mitochondrial substrates such as malate, succinate, obviously fatty acids and protein. And we map these pathways essentially using a technology called extracellular flux analysis. It's a microplate-based technology, so we can run this in a 24 well plate system, a 96 well plate system, or an 8 well plate system. The cells or tissue are cultured within the microplates, and on top of the microplate goes a sensor cartridge. And you've essentially got non-invasive sensors for measuring dissolved oxygen in the media surrounding the cells and the free protons. And the proton measurements, we term this proton efflux rate per, and that is our kind of proxy for glycolytic fluxes and glycolysis. And then the oxygen consumption rate, or the oxygen change over a measurement cycle, can be calculated as oxygen consumption rate, or OCAR. And this is our proxy for mitochondrial respiration. Within the system, there's an integrated drug delivery port system. So this allows you to, in real time, at defined time points throughout the assay, inject different compounds. These could be different substrates. They could be inhibitors of certain pathways. It really can be designed depending on what the nature of your question or research question is, or endpoint goal is. So essentially, the technology lends itself to real-time kinetic measurements. We're using live cell samples, tissues, or even isolated mitochondria in some instances. There's integrated injection ports, so we get real-time effects of things that we can add directly onto the cells and look at their immediate energetic response to those changes. It's label-free sensors, so it's relatively non-invasive for the cells, so they could be used for other purposes downstream, and you get multi-parameter analysis from this. The workflow for a metabolic profiling assay is relatively straightforward. You seed cells in the well plate on days leading up to the assay, unless they're in suspension, and you would put them in the plates on the day of the assay and do pre-treatments in different vessels. you would then pre-treat if they're adherent cells for any time period before you want to run the experiment, or even during running the experiment. We need to prep the sensor cartridge. And the day of the assay, you would prepare the buffer that you're going to use, or the media that you're going to use to run the experiment in, calibrate the cartridge, and run the assay. And then you get your data. And if we think about how we can map sort of energy metabolism from the mitochondria, we typically use a profile which we use in a series of different mitochondrial compounds, essentially, and inhibitors or activators to look at specific aspects of what's going on in the mitochondria of the cells. So this is a typical profile. You have your oxygen consumption rate over time. We take a first few measurements, and that would give rise to our sort of basal respiratory activity. Now, not all of that oxygen consumption is mitochondrial, so we do need to correct for that. I'll come on to that in a little bit. And it's not all coupled to ATP synthesis either. And this is important, and this is where efficiency comes in. So in order to calculate how much of the oxygen consumed is actually driving ATP synthesis and therefore supply for the cell to be able to do the functions or grow, proliferate, we use a compound called oligomycin, which shuts down the ATP synthase in the mitochondrial inner membrane. And that will inhibit oxygen consumption rate to a level that is supplying or to the amount that's supplying the cell with the oxygen that's driving ATP coupled respiration. Now, what's left is a combination of proton leak and non-mitochondrial oxygen consumption. And we can adjust for the non-mitochondrial oxygen consumption by inhibiting mitochondrial oxphos completely using complex 1 and complex 3 inhibitors. Rotaline and antimycin A are typically what we use in the lab. Now, where the trace here with the blue section shoots up, that's where we've uncoupled the cell, essentially. So the reason why cells do not operate, the baseline respiration is not operating where the maximum is typically because ATP demand of a cell controls energy supply and substrate supply. So in order to look at respiratory capacities of a cell, we need to take away that control. And we do this by uncoupling the mitochondria, essentially, and depolarizing the membrane. And then we can get an idea of what the actual capacity of that cell is. So when we do a profile like this, we can get real key information around baseline respiration as coupled to ATP synthesis, proton leak, coupling efficiency, which is essentially the percentage of your respiration that's driving ATP synthesis in the cell. And the more efficient your cell is, the better, in the sense that you're going to use less substrate for the same energy output, and you're also going to potentially use less oxygen in the process as well. Capacity is measured, and we can look at spare capacity by taking our max capacity and subtracting our baseline rate. Additionally, because we're also getting measures of the protons that are being released by the cell, so on the same cells, in the same wells, we're also able to get an idea of what's going on glycolytically. And this is an interesting thing. In most cell types, you're not getting one or the other pathway. Both pathways are active. Both pathways are supplying that cell with energy. And it's not one or the other. And some cells might be more glycolytic. Some cells might be more oxidative. And so this technology lends itself to being able to profile the cells in that way to get an idea of what the requirements are of the cell that you're actually working with. And the main things that we're kind of looking for or interested in, certainly at Biocell Energetics anyway, for our purposes of our clients and things, are looking at, well, what is the baseline glycolytic flux? And how does the cell compensate if you shut down mitochondrial respiration? Does it compensate? And is it flexible to basically adjust its metabolism to support the energy demands of that cell? And then what is the capacity glycolytically of that cell? And that's where the compound at the end there, monensin, comes in. In muscle, it drives an increased energy demand, so we can push So as long as glucose or substrate supply is not limiting, we can in this instance glucose, we would be able to drive maximum glycolytic capacities. So we get three main parameters from looking at proton efflux rate as well, which is our baseline glycolytic fluxes, the compensatory effect, and glycolytic capacity. So I don't have any data on cultivated meat cells, unfortunately, not yet anyway. But in a recent project we're working on at Biocell Energetics is looking at profiling of peripheral blood mononuclear cells. So these are cells these are immune cells of our blood that we take out. And we're interested in looking at what the processing time of the blood by isolating the cells at different times after the blood's been taken, if that has any impact on effect on the energetics of the cells. The importance of this for us was because there's a metric called the bioenergetic health index that's been reported potentially to be able to predict disease in individuals. And if you're sending bloods to clinics to get tested in this way, we wanted to know, well, what's the time frame? How long have we got before the cells become compromised in their function or not? Before we could actually, if you're going to use these types of metrics as an indicator of metabolic health. So we take out the cells, we do excess analysis on them, and what we found is that actually increased processing time, so leaving the blood at just room temperature for longer periods of time, above 24 hours typically, so 48 hours or 72 hours, significantly lowered the rates of respiration, lowered ATP coupled respiration, interestingly increased proton leak, so therefore coupling efficiency is less as well. So these cells are somewhat stressed and they become less efficient. The maximum respiratory capacities become lower, and also the glycolytic fluxes are somewhat lower too. So these cells that are left in the blood for longer are becoming metabolically, or metabolically they are slightly very different to if you isolate those cells fresh or after 24 hours in their energy supply and their function. And what we're able to do also is take our ATP linked oxygen consumption rate and our baseline glycolytic rates and convert these into actually estimated ATP supply fluxes. So then we can present data from oxidative ATP over glycolytic ATP and look at what the glycolytic index is of a cell, essentially. So anything that kind of where we've got the oxidative over glycolytic energy map here, anything that falls in the blue shaded area would be classified as having a glycolytic index of over 50%. So that means that 50% or more of its energy supply is coming from glycolysis, and the rest would be oxidative. If it falls to the other side, then it's predominantly oxidative. So it can also give you indication of, well, what's the cell's requirements here? How they function and what do they need to be able to sustain them and give them the energy that they need. In this instance, we were then able to predict that because their metabolic function was affected under these sort of quiescent conditions, that if they were activated, would they be able to meet the energetic challenges of activating an immune system? And we did this and we mapped total energy supply in the before and after activating the cells. And we see that the difference in meeting those energy challenges for activation is also somewhat compromised in the cells that we isolated at 48 and 72 hours. So our conclusions are that essentially by doing the profiling of the cells in this way, we were able to conclude that the mitochondrial function becomes less healthy, more unhealthy at 48 and 72 hours, and your glycolytic phenotypes are becoming more abnormal than normal. So thinking about how we could use this, if you were to look at different cells, for example, and this is just an example mock set of data I've put together, where if you were to look at cell A versus cell B versus cell C, clearly their energetic phenotypes are different from cell A, predominantly oxidative, cell C, predominantly glycolytic. So their needs and requirements in terms of substrates and the byproducts that they will create are going to be very different. So mapping the energy fluxes of cells in this way can give you information and inform those types of decisions that you might want to know about before you go and scale up your kind of cell stocks. But essentially, what's also quite interesting is that some cells are quite metabolically flexible. So if you were to shut down a pathway, say in cell A, you shut down mitochondrial OXPHOS. Actually, because under maximum conditions, it has quite a large glycolytic reserve, it would be able to sustain its energy demands by compensating glycolytically. And if you calculate this, we term this a supply flexibility index. So a cell A, for example, that has 100% supply flexibility index means that it can shift between different metabolic pathways to sustain its energy demand, stop it from dying, make sure that it continues to grow and proliferate. Cell B is not so flexible at 75%, and cell C would be 40%. So it's more dependent on a specific pathway for its energy. So if that becomes compromised, or if things become rate limiting in that pathway, you're going to get a drop in ATP supply, you're going to get a drop in function, and ultimately a drop in growth rate and probably cell death. So this has many applications in many different fields, as I've already kind of addressed the markets that we kind of support. But I'm really intrigued to know how it could link in terms of profiling cells, particularly that are used for cultivated meat, and also what effects some different media compositions having on the cellular energy needs and requirements of a cell, and whether you can use energy mapping in a way to be able to fine-tune media composition and inform sort of cell selections that you might want to move forward with when you go to more mid-stage pilot scale, or even large late stage scale up. But I also think you could use profiling at the different stages as well. If you're able to get cells out of your tanks and fermentation tanks, they might be they'll be in suspension, but you can still profile suspension cells using this technology. So if you had this kind of setup in your lab, then you'd be able to kind of take some cells out and actually profile them within a few hours and see whether energetically they are as normal as they would at sort of a lower scale or when they start, and get an idea of whether their energy demands or capacities are changing, whether they would classified as healthy or abnormal or normal. So I think there's many opportunities potentially with this technology to be implemented at different stages, particularly around cell line development. Could it potentially identify bioenergetic markers that then later correlate with high quality yields and consistent tissue formation? Around media composition, how does adjusting, say, glucose, or how does adjusting moving away from animal serums to plant-based or alternative proteins in the media, how is that affecting the cellular energy fluxes in cells, and what does that mean for scaling up later down the line? And scaffolding as well. So when you start bringing the cells together and scaffold them, how does that change their energy requirements? And it becomes a little bit more tricky to profile 3D tissues, but it is possible. But there's many different opportunities, I think, in the cultivated meat industry where this technology might be able to support some of the questions around the solutions or technological issues and problems and bottlenecks that you're facing. So kind of like a mock example, if you took a chicken, a cow, and a pig, it's likely that you're going to get different metabolic phenotypes and different profiles. And ultimately, if you go through the process of the data analysis, eventually you can get that sort of ATP flux data at the end and get an idea of what the cell's requirements are and how efficient they are. And this maybe is more insightful maybe when you're looking at different media compositions and growing cells in different medias and things, and how that is potentially changing energy flux. or if you've got a cell line that you're not really sure what its main requirements are. Is it more glycolytic? Is it more oxidative? Is it dependent on specific substrates? You can start asking these sorts of questions early on, and then maybe even tailor make your media composition around the cell line that you choose to use for cultivated meats. So the key takeaways really is that this technology can be used to profile energy pathways and capacities in cells and tissues. It may be able to help improve efficiencies in cultivated meat production by understanding the cellular energy demands and capacities of a cell, and how media potentially impacts the growth and can inform cell line selection, particularly when we're talking about scale up and product consistency, if you were to be able to profile cells as they go through the supply chain. So looking ahead, I think some refined metabolic profiling might help to drive breakthroughs in cellular agriculture, certainly early on with cell line selection. I also think there's probably cross-industry potential here as cell lines are developed for cultivated meat. Can they be used in other fields as models to test things like environmental toxins or even drug compounds? And could alternative proteins and alternative medias also produce a more sustainable option for routine cell culture and modeling in research for basic research studies? And I'd be really intrigued to know whether anyone's thinking of that outside of cultivated meat. Can we use these medias to actually just grow cells for research purposes as well? And is that more sustainable because you're not using meat-based bovine serum? So I'll be really really interested to talk to anyone if you're kind of interested in learning more or talking to me about this, or if anyone's got any questions, more than happy to take any questions today. And my contact details are there, and our website if you want to learn more a bit about Biocell Energetics is also detailed on this slide. That's that's the end of my talk. So thank you for listening.
Excellent. Thank you so much. Have time for some quick questions. Excellent. Well, we are at the top of the hour. This was interesting, certainly really foundational stuff, I think, in the energy side of things to figure out that will dictate quite a bit around cost and process efficiency. We've been thinking about that a lot actually for our modeling study. So we might, reach out to you, Johnny, if we have questions on some of these things or good data sources or whatnot. But yeah, thank you again for presenting. Good to see everyone, and have a good holiday and New Year.